Each registration is unique and grants the right for the individual registrant only to view this webinar. Sharing this webinar with unregistered viewers either during or after its performance violates Pharmatech Associates copyright and is prohibited. If you've not already done so, we ask that you take some time to read the Pharmatech Associates course disclaimer. You can download this presentation, including the disclaimer, from your USP Education account. Please follow the instructions in your confirmation email to download the presentation. Welcome to today's USP and Pharmatech webinar, Biosimilars Meeting the FDA with Your Biosimilar Product. Thank you for taking time out of your busy schedules to join us. Presenting today is Dr. Claudia Lin, Executive VP Biologics Lead at Pharmatech Associates. Welcome and thank you. I will now hand the presentation over to Dr. Lin.
Thank you, Sylvia, and thank you all for joining us on this webinar today. We're talking about biosimilars, preparing for your FDA meeting. Next. What we're going to cover is as follows, and Pharmatech Associates have helped over a decade now biosimilar companies around the globe to advance their biosimilar development programs and getting approvals from health authorities around the globe, not just the FDA. So we would like to help you to get there. Next. Pharmatech Associates is the consultant arm of US Pharmacopoeia. And I hope all of you are very familiar with United States Pharmacopoeia, its missions, and its gold standards for pharmaceutical manufacturing and quality. And we are the consulting arm for US Pharmacopoeia. Next. Today, in the next 40 minutes or so, I'm going to present to you the development pathway and the data packages, as well as the strategy for meeting FDA requirements for your biosimilar programs. Before we get there, there are two important documents or laws that we all must be very familiar with. One is the Biologics Price Competition Innovation Act, BPCI Act. This is the previous slide, but it's okay. I'm going to talk you through it. So BPCI Act officially started the journey of FDA's approval for biosimilars in the US. It was approved and signed into law in 2010. BPCI, therefore, is fundamental for all of us to learn and understand what it is and what it is not. And what it does not allow for lowering standards for drug approval. It does allow for abbreviated pathway on demonstration of biosimilarity to reference products. It is not the same as 505(b)(2), which is quite often a confusion amongst people who are involved. But it does permit a less costly drug development program. And the other important thing for us to always be familiar with are the series of FDA guideline documents, and they've been publishing consistently that describes the expectations for scientific considerations, quality considerations, clinical pharmacology data guidance, and then now post-approval manufacturing changes, and then how to demonstrate interchangeability, and then most recently, a guidance document that describes the trend to phase out animal studies as a requirement for your biosimilar program, especially with regard to monoclonal antibodies. So these are the laws and guideline documents that we must be always following and get ourselves very familiar with. So I list this list of guideline documents on the next slide. And we can skip now. I'm sure this is a reference for all of you, and you have them in your hands to refer to whenever you have a specific topic. Next slide. So let me start by describing to you a strategy for developing your biosimilar. And there are two very critical words to remember. One is stepwise. And I tried to illustrate what that means on this slide in a pyramid where you have different layers, levels, sequence that you must accomplish before you can be sure that you have the data package you need to apply for a biosimilar approval for the FDA. So the stepwise approach indicates that you need to start with preclinical analytical characterization. And that data package, if it shows initial biosimilarity, then you're ready to manufacture your biosimilar product for going into clinical trials. And that clinical lot needs to be demonstrated to show the biosimilarity. And these two are the in vitro studies that's required. So prepare for preclinical analytical characterization data package, and then get your biosimilar manufactured, ready for clinical study. And then those lots need to demonstrate biosimilarity in vitro. Once you have that, the next step is considering what non-clinical studies and clinical studies you're going to perform. And non-clinical studies usually are animal studies that demonstrate PK, PD, and toxicology profile, and that needs to look similar to the reference standard products that you're trying to generate the biosimilar product to. And then based on those studies, you may need to go into just a brief phase 1-2 clinical pharmacology and PK/PD studies, or you need to go more advanced to a phase 3 to demonstrate comparability in efficacy study. So this stepwise approach is very important for you guys to keep in mind, and then really perform them according to the layers of complexity that you introduce to the data package that you will be generating. And however, the other key word to remember is to achieve totality of evidence. So stepwise in your design of your program and performance of your studies, but what you want to achieve and what FDA is looking for is a totality of evidence. So next slide, let's talk about that, what that means. Totality of evidence means FDA is looking to see if combining all the steps and data packages that you present to them, do they see a totality of evidence that supports the approval? There is no one size fits all to attain this biosimilarity and approval by the FDA. Their assessment of each of the program is case by case. However, there are some general rules for how they would view your data package in order to make their decisions. And what you guys are supposed to do is to figure out how to define your critical quality attributes based on risks. And normally there are 3 tiers of quality attributes. Tier 1 are the most critical ones, where you must demonstrate biosimilarity, and these are things like potency, for example. And there are other critical quality attributes which are highly important because they're high risk CQAs, so you must demonstrate similarity. Tier 2 are the ones which are median risk, and therefore what you need to demonstrate is an acceptable range. And in your analytical package, these are deemed to be tier 2. Then there are tier 3 ones, which are known lower risk attributes. For example, you have a species of oxidation and others, and you need to understand they exist, and you need to have a quality risk assessment to show why they are lower risk than the tier 1 and tier 2s. So with the 3 tiers, you have a good profile of the quality attributes which need to be absolutely demonstrated to be biosimilar to the reference standards, or you have a group of tier 2s which are acceptable as long as you maintain certain ranges, and then you have the tier 3s which are known, but they don't need to be tracked as rigorously as tier 1 and tier 2. So having said that, the system from which you perform all these studies is very important to show that all the studies are done under a compliance and under control system. And we're talking about your quality system. For biosimilars, there's quite often a confusion about how rigorous you need to maintain your GMP quality system. And in fact, I think GMP compliance is more important for biosimilar programs than the novel biologics or novel therapeutics, because you really need to make sure from very early stage of your clinical studies that your systems are robust enough to give you the results which are dependable, reliable, and also you can trust these data going forward to the product launches. So we usually emphasize a phase-appropriate GMP quality system, and these need to be demonstrated to be appropriate for the stage of your biosimilar development, and therefore coupling the CQA considerations and your quality system, you have data packages which are defendable to the FDA. Next. And then let's talk about analytical data packages. Usually for a typical biosimilar, you have the data packages which compose of the 4 different aspects of the characterizations. One is primary structure, and that's the amino acid sequence. And then 2 is the higher order structure, which are secondary and tertiary structures, which are also important for the safety and functionality of your biosimilar product. And then you have purity and impurities. This is a large group of characterized attributes that demonstrates either the purity or the impurities, which are important to be in the acceptable range. And these are things that include charge variants, host cell protein, host cell DNA, and then we also have process-related impurities, such as protein A, such as anti-foam. It is really a long list of quality attributes that you need to pay attention to in order to demonstrate the appropriate level of purity and also acceptable levels of the impurities. And lastly, but very critically, the biological activity in potency assays. So these are the analytical data packages usually composed of. And then next slide, we're going to talk about the key points for these analytical packages as a recap. Really, the most important thing is to define your critical quality attributes as early as possible. Why is that? Because you really need to know what you're driving at achieving from early on of the development program. And so you can really tailor your process development and process control and also QC lot release criteria according to the defined CQA. So don't wait until later in the clinical development program to define your CQA. You're a biosimilar, so you already have a list of critical attributes you know based on your knowledge of the reference product. These are the CQAs. So define these early for you, but also define the CQAs which may be different for your biosimilar product versus the reference standard product. And then drive process development, process control, and lot release decisions based on that. And the other very important aspect is as you are figuring this out, and you really make sure you need to have a communication strategy with the FDA on how you're thinking about demonstrating the similarity, and then the data package that support your arguments. So along the way of figuring out your development strategy, you need to also think about your communication strategy with the FDA. Next slide. So I cannot overemphasize the importance of analytical methods in a biosimilar development program. So let's focus on that a little bit more. So naturally, we need to consider the method sensitivity, the method specificity. A question that often comes up from our clients is how much and when the methods need to be validated. And this is a very good question and an important one for biosimilar development, which may be different from novel biologics development. When you have a biosimilar and you need to demonstrate similarity, you have a shortened development timeline in terms of the clinical programs that it's required. Therefore, you need to make sure that the methods are really reliably telling you what your process is giving you, as well as what your product is able to do or not able to do, both in vitro and in vivo. Therefore, the validation of these methods becomes more critical from earlier on than later compared to a novel biologics program. And then you really have to answer this for yourself as for a specific method. When I need to have a full validation, when do I need to expand the validation? When, for example, your manufacturing is extending beyond the primary site of manufacturing. And also, when you have a method that can demonstrate multiple quality attributes, that requirement for validation becomes even more important and more stringent. So above and beyond the sensitivity and specificity, validation of analytical methods for your biosimilar programs becomes a very strategic decision for each company, for each of your program. And there is great advantage to having methods which you can totally trust, which may serve multiple purposes. For example, single assay used for your PK and also for immunogenicity testing. And that assay becomes very critical, and then you really want to make sure it's fully validated before you use it to draw conclusions on PK and immunogenicity. Next. So you may have heard one of the latest guideline documents from the FDA is indicating that they are phasing out the requirements for animal studies. And so although this might come as a relief to a lot of us, we also need to be very careful in how we act on this potential trend from the FDA. Because despite of this guideline documents, it will take time for even FDA to figure out how to actually phase out the animal study requirements over time. Therefore, as a developer, it is important for you to assess and build in the animal data requirements into your program timelines so you don't have an unrealistic expectation by skipping it at the offset. And then the other thing I already mentioned a few slides earlier is to make sure your quality system is in a good shape so that not only you can trust the data coming out of your quality system, the products being produced under your GMP quality system, but also you have the data that is robust enough to demonstrate to the FDA in your data packages, in also the upcoming inspections, And so quality system is not something that you should think afterwards, but really ahead of the whole plan. And so animal study or quality system, these are things which are flexible in the strict guideline documents, but nevertheless, a developer has to really consider how you can build in the timeline and requirements, as well as the appropriate level of GMP readiness for your biosimilar programs. And then FDA ultimately has the discretion to list animal study data as a requirement for your biosimilar program. And again, the totality of evidence indicates that they would assess your program on a case-by-case basis in order to judge whether the totality of evidence is reached. Next. So now that I've illustrated to you a strategy for developing your biosimilar programs, I like to talk about the interactions with the FDA. So preparing for FDA interactions is an ongoing process, and roughly it's divided into 3 stages. One is pre-IND, where you are preparing for a submission to IND. And then there are meetings that requires preparation, data packages, make sure that you indicate studies which are in progress, not just studies which are done, because FDA really wants to see that even though you don't have everything that's required, but you have thought about everything. And then the communication is very important. It needs to be done ahead of time, and then giving them enough time to review your briefing packages. And then any action items from reviewing the briefing packages can be quickly followed up before you submit your IND. And then between submission of IND to developing your programs to further stages, you're going to have more planning to do. So the animal studies, whether or not they would require it, in fact, or require more of what you've done. And then very importantly, what kind of human studies are required. And then are you going for the interchangeability status, or you are happy to reach just biosimilar status? And what would be the development activities involved in order to achieve either or both? So these are the additional development planning work that needs to happen. And then this is in response to the feedback from the FDA. And the third stage, which is the final stage, is BLA submission. So at that point, all your data packages are complete. All the process and analytical methods are fully validated, and you can demonstrate compliance under which all these studies have been performed. Therefore, you're ready for an on-site inspection. So these are the 3 stages of interaction with FDA. Next, and they're usually conducted in a series of meetings. Next slide. And these meetings are very standard, and I'm sure the regulatory affair colleagues that your company has can guide you through all these different meetings. So I'm just listing here for your reference. And the overall planning is what I described in the previous slide, and it really is pre-IND becoming really the critical one that set the tone for how FDA is going to view your program and whether all the planning you're doing is sufficient. And then you do all the studies in between, and then you submit BLA. So the meetings are very important, and the communication, even when things don't go as planned, you need to have communication with the FDA instead of avoiding it. Next slide. So now I'd like to talk about some of the challenges we have seen in working with clients, biosimilar development companies all around the globe. Even though this talk is focused on FDA, these challenges are really common challenges. So we would like to just call your attention to some of them. We have described a stepwise strategy. And if you have a strategy, but you fail to execute, obviously, that's a challenge. But more specifically, the reference product selection has been a very challenging issue, especially for companies or regions in the world who are just getting into the biosimilar field. And this took quite a long time for the industry to understand how to select reference products in order for that reference data package to be representative in the eyes of the health authorities. Another common challenge is on your specification. Even though you know what you're trying to make your product to look similar to the reference product, it is difficult to just get the specification for that reference standards and apply it directly to your biosimilars, especially early on in your program development, for example, R&D stage. Because your process are relatively immature, it is unrealistic to set the specification according to a mature commercial product. And therefore, how do you do that? And how do you actually justify a specification? And how do you evolve that specification along the different stages of your development? This is a common challenge. With more and more companies from different parts of the world actually getting their biosimilars approved, comes the challenge of post-approval activities. First are the post-approval commitments that you submitted during your BLA, and how do you make sure that you actually carry them out to fulfill these commitments? And then one particular common scenario that comes about post-approval is changes in manufacturing. And this could be manufacturing scale changes. This could be manufacturing site changes. This could even be some changes in some critical operational steps. So how do you get that managed and continue to get approval for your biosimilar from the FDA and from other health authorities? These are very common challenges from biosimilar developers all around the world. And then next slide, I also like to focus a little bit on the difference between US FDA and EMA on their regulation on biosimilars. So I would say over the last decade or so, FDA and EMA are converging more rather than diverging from their regulations on biosimilars, which is a good thing for the biosimilar development world around the globe. And however, there are still meaningful differences. And I listed in this table some of them. For example, the definition for biosimilar. FDA says with no meaningful differences. And EMA regulation says similar in safety and efficacy. So you can see essentially they are the same. However, the FDA definition does allow you more flexibility to explain when you see a difference. Why is that meaningful? Why is that not meaningful? Another market difference is the access. When you achieved a biosimilar, the US, especially if you've reached the holy grail of achieving interchangeability status, for FDA, it only allows you to do so within the entire United States. But in EU, you have different countries. When you achieve interchangeability in one, it does not mean you have achieved that in the entire EU. So these are meaningful differences, and there are also nuances in how FDA versus EMA evaluates a biosimilar program. So this is just a list of areas that you should pay particular attention to. Okay, so the next slide, I'm listing typical analytical data package for a biosimilar of antibody, monoclonal antibody. And you probably have already uh, established tests for all these different quality attributes. And this follows the strategy that I mentioned, is defining your CQA early and then developing analytical methods which reliably demonstrates the performance of your process and your product, and therefore justifying your biosimilarity in vitro as well as in vivo. So I've concluded my talk at this point, and thank you very much for your attention. Now we have some time to go into question and answer. And as we are taking the questions and answers, we might not be able to get to all of them today, but if we don't get to your questions, we will get back to you in writing in the next few days. Okay, so thank you very much for submitting your questions already. So let me go to the first one. Is BPCI relevant to human health and animal health, or just human health? It's relevant for both. How do you make sure the potency assays are adequate for BLA? That's a very, very important question. And it goes to what I've mentioned a little bit earlier in my talk, which is potency assays are the ultimate standards for demonstrating biosimilarity. However, the assays that you use for your biosimilar may be different from the assays that the reference product uses. So how do you make sure that what you're demonstrating in your assay is adequate? Again, it goes back to two things. One is, does this assay reflect the mechanism of action? So you have to justify why your chosen assay actually would reflect the mechanism of action for your biosimilar. And if the answer is yes, it does reflect MOA, then the next question is, your assay performance is adequate for carrying this product to an approval stage. So what that means is, we talk about the assay sensitivity, we talk about assay specificity, and then there's the robustness, the whole ICHQ validation criterias are met. And then in addition to that, you have to show potency assays can adequately reflect manufacturing variabilities. So if your potency assays are so variable that even when you're manufacturing a product that is out of the range, but because your potency assays are so variable that you cannot really tell whether it's truly 80% to 120%, or at least 75% to 125%, if your assay is just too variable, then it is not adequate to be used as the potency assay for your commercial product. So yeah, potency assay is very, very critical, and it's one of the more difficult tasks to achieve in a biosimilar program for the reasons that I mentioned. Next question. Do we have to use fully validated analytical methods for our biosimilar characterization? The answer is not necessarily. And this is because you have the release assays, and testing for those have to be using validated methods. But if it's just for characterization, you are not required to use fully validated methods. However, I also mentioned the more validated these methods are, the more confident you are in being able to characterize your biosimilar in a way that will provide not only to yourself, but also to the FDA, the totality of evidence. So it is recommended that you validate assays as early as possible, and you validate all the assays you need for release testing. And then for the characterization testing, these assays need to be validated for intended use. And that intended use is defined by the stage of the program, the criticality of that quality attributes, and then also the analytical operations in your company. For example, if all the products are tested in one QC lab, then the qualification of that method is relatively easy. But if you are going to have multiple QC labs doing these testing, then that method needs to be validated across the different laboratories. So different levels of validation for the assay needs to take place. Okay, so next question. Can an applicant demonstrate that a biological product is biosimilar and interchangeable using one clinical study? Right. Okay. So it is possible that you could achieve that. But going back to the stepwise approach, and if FDA would consider all your data package, in this case, one clinical study, along with preclinical animal studies, along with your in vitro analytical comparability studies, they would decide whether the data package combined is sufficient. So I would say it is possible you could just do one clinical study. However, it really depends on all your other studies and results from those, as well as whether you can demonstrate the control under which these data are achieved. What parameters to be studied, it very much depends. So it depends on the biosimilar product, depends on other evidence to contribute to the totality. And so it's difficult for me to specify which parameters to be studied. But the most basic thing is PK/PD, as we all know. Next question. What are the challenges about developing appropriate potency tests to be used for comparison purposes with the reference product? I believe I covered this in answering the first question already. So to recap, it is demonstration of the potency assay is going to represent the MOA of your biosimilar, and also the assay itself needs to meet the criteria for being a validated and viable assay that properly reflects the variability of your manufacturing as well as the activity of your product. Next question. If the formulation is similar to a patent product, is in vivo study still mandatory? When can animal studies be exempted? Yeah. So if the formulation is similar to a reference standard product, it is possible that you would not need in vivo animal study. However, you cannot assume that's the case. Just because your formulation is similar, you still have to demonstrate in your own hands the similarity at in vitro level under a well-controlled quality system, and then FDA will judge if the totality of evidence is achieved. So I would not assume just because the formulation is similar, therefore you would not need to be doing animal studies. Okay, so let me go to, okay, so please provide reference to the recent release from FDA about phasing out animal studies. Go to the latest guideline documents from FDA, and it's there, the most recent guideline documents for biosimilars. Just go to FDA website and you'll see that. Next question. Do you believe it's possible to defend a change in this master cell bank with not providing new in vivo data with the comparator, with robust analytical comparability results and PK comparability with previous cell bank? Yeah, I'm really glad you brought up this question, and therefore the importance of master cell banks. So master cell banks are really the starting point of your product in the eyes of the regulators. And then if you change anything to the master cell bank, then in theory, you have to justify why this now would still result in the same product that you originally were developing. So if a change in the master cell bank is something that would not result in significant, meaningful changes that will affect CQA, there is a chance you can get away with not doing in vivo data. But it really depends on what that change is and how much data you can generate to demonstrate the fact that this change does not result in meaningful changes in critical quality attributes. And these critical quality attributes will then affect the safety and efficacy of your product. So I cannot give a general answer, but I would say it is possible to not have to do in vivo studies with the master cell bank change, but it really depends on what the change is and what kind of data package you can put together to demonstrate this change does not yield meaningful differences in the CQA. How many batches of biosimilar reference product must be studied for evidence of biosimilarity? Yeah, so this is a question that early adopters of biosimilar development often ask. And there were many different answers to this, and also depending on where you are in the world, the health authorities will have different answers to you. So the general answer is it really depends on what constitutes a statistically significant number of lots, because that is what you need to strive at, is to say, I am sorry, I'm demonstrating how the reference product is behaving based on characterizing, testing all these different lots. And why do I believe all these different lots together is representative in a statistically significant way of what the reference standard product looks like? So it's a statistical question that you have to answer and you have to justify. And also the different lots may come from different regions of the world, and it may come from different periods of different process, versions of the processes. So there are a lot of complexities that you need to look into before you say 12 lots is enough, 18 lots is enough. Okay, next question. In the case where a company wants to replace one of its extracted protein APIs by a recombinant version, does this automatically require a new NDA and NADA? Okay, I have to think. So this is not a recombinant. So it's extraction versus a recombinant. I believe this constitutes a very significant change because everything is different. Even though in the end you're trying to make the same product, but where it came from the beginning is different. So I would assume this is going to require a new NDA. Next question, why USP made the acquisition of Pharmatech Associates? Excellent question. I'm happy to answer that. So USP has been an institution for about 200 years, providing standards in terms of procedures, test methods, as well as actual physical standards for the pharmaceutical industry. And by doing so, it is a non-biased institution to the pharmaceutical industry. And they also have arms that focuses on advocacy for using these standards, but also for maintaining a high level of quality and compliance for pharmaceutical industry. So USP made the acquisition of Pharmatech Associates so that we can work directly with industry to advance the advocacy aspect of the USP mission. And Pharmatech Associates, being a 30-year-old consultancy in the US, had established ourselves to be a leader in the space of pharmaceutical development, especially with our expertise on CMC, quality, regulatory, biosimilar, and novel product development strategies. We were very, very fortunate to be the target for USP, to have acquired us to be the consulting arm for USP. Do you always need to generate comparative data with the reference product when CMC changes are made in the post-approval life cycle of a biosimilar? Are there exceptions? So I would reference the ICH guideline for post-approval changes, and this applies to biosimilar as well. Whether or not the CMC changes is deemed to be significant, whether it's changing the established criteria or not. So I would refer to the ICH guideline for post-approval CMC changes. If USP monograph has a very wide limit of assay, difference is more than 70% between lower and upper limits, how can a clinical study be justified with respect to dosing to patients, which is calculated based on the assay? Yeah, that's a very, very good, I think, almost philosophical question, and I would give my personal opinion on that. USP monograph establishes a limit, and that limit is something that USP can not only require, but expect if the industry performs the assay according to USP. It does not mean the company should not achieve, say, a higher robustness or a tighter specification in their particular test if they deem that test to review something that requires a tighter control. So it is general standards by the USP. It's the minimal you need to achieve if you follow the monograph, and it's achievable by USP practices. However, when you use that for your particular testing for your product, you would understand if a tighter limit, a tighter control is required. And then it is up to you to demonstrate that you can achieve that tighter control in order for this test to be meaningful indication in the clinic. Okay, next question. When scaling up manufacturing from laboratory to commercial production, is it acceptable to consider consistency not to be an issue if the CPPs remain within the control range and the CQAs are confirmed to be equivalent using analytical methods? And my simple answer is yes. But of course, that yes is qualified by whether or not everything is equivalent in terms of your testing conditions, and also whether your methods are truly reliable to indicate that the CQAs are confirmed to be equivalent. And it's really good to see that the CPPs can remain within the control range from laboratory to commercial. That's excellent. That probably shows your ability to do a very good DOE so that you can scale up very successfully by the demonstration of a well-controlled CPP. But I would still caution CQA is demonstrated by your analytical methods, and you have to make sure the analytical methods are actually equivalent or equally robust from laboratory stage to commercial stage. Next question, if we're manufacturing biologics for phase 1 clinical trials, do we need to follow GMP? Yeah, so I touched on this briefly. I would say phase 1 clinical trial is going to human, and GMP governs GMP is the governance for manufacturing for human studies, and therefore you do need to follow GMP. Having said that, GMP is the law, and quality system of your company interprets how the law is going to be implemented in your company. So usually for phase 1 production, the GMP requirements translated to quality systems would focus on ensuring safety. So yes, you do need to perform under GMP. However, the elements of GMP as defined in your quality system for phase 1 needs to really make sure the safety is ensured. Whereas for later phase, you also have to make sure the efficacy and robustness of the manufacturing process are both also ensured. So there's a difference in the standards, but yeah, you do need to follow GMP even for phase 1. Can a reference product be used as a reference standard for biosimilar product development and for the analytical method validation before the 2-tiered reference standard system is set up? Yes, we've helped clients in the early stages of their development program, and that's exactly what we advise that they do. So the answer is yes. What unique benefits, product, services offer? What unique benefits, product, services offer that others do not? I'm not sure what this is referring to. If you're asking about Pharmatech Associates, I would say, first of all, Pharmatech Associate has a 30-year experience, track record of being a leading consultancy in the US for the pharmaceutical industry. Our expertise is comprehensive, not just in biologics, but in all pharmaceuticals. And then with the acquisition of USP, we also have a much broader global view, and we're helping markets, not just in the US, but also ex-US, especially those who are trying to develop products to be approved by the FDA. And so comprehensive, proven track record, USP, advocacy for USP, and the global view.